Thank you so much, Evita, for having me here, and thanks everyone for coming. I know it's sort of the end of the semester, so I'm happy that it's not just the two of us and an extra person in the audience. So today I will talk about applications of topology. So this is like a very general introductory talk based in a big part on lecture notes by Gunnar Carlson, who was my PhD supervisor when I was at Stanford. But then I try to give reference when I give like some examples of applications, so that if anyone is interested, you can take a look later. So how are we going to apply topology? So what I like to do in my free time is apply topology to data. And data is often given in the form of point clouds in Euclidean space. And for a mathematician, a point cloud is a finite metric space. And we often have problems analyzing data because it's given, say, in the form of very long vectors. So imagine, so one data set the data set that I will show later is going to be with gene expression data. So imagine that you have like a patient and they have a lot of genes in their body, and then but what is basically done is that you can represent this patient with a vector where every entry is one gene, and then you have the number that says how this gene is expressed. But then also imagine that, for example, if you are interested, say, in determining which genes play a role with breast cancer, not all genes are going to be relevant. So that means that what often happens is that the data collected is very high dimensional, but probably the sort of intrinsic space that you're interested in is actually low dimensional, but you just don't have access to it. And then what often happens is also that the data is noisy. So here you can think of internet, like when you click on Google, Google is collecting data so that it can suggest plane tickets, umbrellas, like nice shoes. And sometimes you click something by accident, so that would be noise. And then the task is how do you separate noise from like the underlying truth in your data. And the goal of topological data analysis, or applied topology, if you wish to call it like that, is to use the machinery from algebraic topology to develop tools for data analysis, to sort of study qualitative features of data. And now, when I say, so then I guess there's sort of a famous citation, data has shape and shape has meaning. So maybe let's just see what sort of kind of shapes of data like we might be interested in. So over here, so the data would be the blue dots that you see, and it looks roughly like these dots lie along a line, and this is a problem that is very well studied already, namely in statistics, and sort of the answer to, like, so how do you model such data? Well, you use linear regression. And then another possibility is that you have data and you want to divide it into groups. So for example, I don't know if I say, again, breast cancer, because that's going to be an example that comes up later. If you have gene expression data coming from patients, does someone have breast cancer or not? So maybe you would want to group the data into two groups. Then what you're searching for in your data is the equivalent of connected components in topology, and these are called clusters. And clusters are also a very well studied, so how to clustering is also very well studied in computer science. But not all data lies along linear subspace, and not all data can be broken up into groups as nicely as on the previous slide. And sometimes it happens that data has slightly more interesting shape. So for example, over here, so this is an example where now the screen is slightly squished, like to be adjusted. Instead of a sample from a circle, we have a little ellipse. But what we want to capture in this data, for example, would be the loopy behavior of the data, the loop. And this is something that does occur. So for example, if one observes the populations of prey and predator. So you have a population of bunnies and foxes, and then when there are a lot of bunnies, that means a lot of food for the foxes, so the population of foxes goes up. But then, of course, because they eat more, the population of bunnies drops, and then there's no food, so then the population of foxes drops, and it's a cycle. And so if you plot these populations, what you see is cyclical behavior. So there are data sets where we are interested either in sort of if the data is loopy or if it's, for example, periodical. So that would be an example where we would want to say something about loops. And then another very common thing that can happen is that we are looking at that we have a single cluster, but that cluster has interesting shape. So for example, over here, we have the central region where you see the blue dots, and then you have two flares sticking out. And then, so this is an interesting shape of a cluster, and then the question would be, is there sort of a difference between, say, the data points that lie on one of the tendrils or between those that lie in the central region? And of course, probably here in the audience, we all know that topology is the branch of math that studies shape. And there are three reasons why we would like to use topology to analyze these types of data sets. So the first one is invariance under deformation, coordinate freeness, and compressed representations. And now I'm going to elaborate a little bit on each of these. So here we're in the situation of a very famous mathematical joke that a topologist is a mathematician that cannot distinguish between a donut and a coffee cup. And my advisor would always say that saying that, you basically shoot yourself in the foot, because who would want to hire someone that cannot distinguish between a donut and a coffee cup? And it's a valid point. But then we would like to sort of stress the positive aspects of having this deformation invariance. So namely, if you go to a basketball game and you see banners, even though the fonts are different, you can still recognize the shape of the letters. And this is sort of, and when viewed in that light, this invariance under deformation can be strong. Mm-hmm.
Can you say a little bit more about what you mean, invariance under deformation? In other words, what are you talking about when you talk about invariance? Patterns? Is it like patterns?
No, it's more like homotopy equivalence or like homeomorphisms.
Of what, though?
So, I mean, well, here in this particular case, like you would have that the top A is homotopy equivalent to the bottom A and has the same homology groups.
Yeah, but what is invariant? I mean, your data set is not a space.
So
it has a linear structure.
That's right. So we are not in the data set setting just yet. So right now we are still in desirable properties from pure topology.
Of what?
Topological spaces.
Oh, okay.
Yeah, so this is still...
Topological spaces.
That's right, yeah. So right now I'm like, why is topology good? And yeah, the object we study...
Eventually you're going to be most interested in point sets and then edges connecting those and perhaps spaces connecting triples and...
Exactly.
Things. And
yes.
So you're basically going to be thinking about simplexes that are joined together. They're topological.
That's right, yeah. Even though today I'm not going to talk about simplices just because I'm going to focus more on showing you the methods and the ideas behind it. But yeah, that in practice, that's exactly what you would do. But yeah, so basically what we're now looking is just why we think that topology would be good to use in this setting. So then coordinate freeness sort of is related to the problem that I mentioned very early on, like for example with gene expression data and very high dimensions. So with like in topology, we study properties of objects, for example, loops, connected components, and it doesn't matter how they're embedded in Euclidean spaces. So that is sort of what is meant by coordinate freeness. And then finally, one thing that topologists have worked a lot on is finding compressed representations of data. And this is sort of where what you mentioned comes into play. So we start with a continuous object like a circle, but what we want to do is we want to find a nice combinatorial representation, say approximate this with a graph or with a higher dimensional simplicial complex. And And then we have two tasks, which are sort of a similar tasks to what we have in topology. So one is we want to be able to measure shape of, so in topology of topological spaces in our setting of data sets, and we want to be able to represent shape in the sense of in topological spaces with combinatorial objects, and in data, maybe very high dimensional data sets with lower dimensional combinatorial representations like graphs. So what I will do now is I'm going to give you an overview of two most popular ways of addressing each of these. And we're going to start with representing shape. A very popular way to represent the shape of data sets is an algorithm called Mapper. And this one, so Mapper was developed by Gurjit Singh, Facundo Memoli, and Gunnar Carlson. It's actually, so it's called a data discovery tool in the sense that it helps, like you get a visual representation, low dimensional representation of your data set, and the idea is that you can use that to find, say, subtypes of cancer or, so that's going to be the examples that I give, but basically interesting subsets, parts of your data set. And this was also also like one of the first startups in topological data analysis. It's actually my advisor like retired from Stanford so that he could be a part of this company called IASD, where they worked on improving Mapper and sort of the user friendliness of the algorithm. And I'm just going to give you a very basic idea of how it works. So we start with a covering. So over here we have 3 sets in the covering. So the turquoise set, the blue, and then 2 blue, like dark blue and light blue. So the idea is, if we have this covering, actually, so maybe now is the time that I would use chalk a little bit. So just let me slow this down. Okay, so the green and the yellow are not very easy to distinguish. So let me put purple over here. Okay, so we start, so covering again is like in pure topology, but like you can imagine that you cover your space with some sets. And then the idea is that, so the goal is that we want to approximate this with a graph or with a higher dimensional simplicial complex. And the way we do this is that to each set of the covering, each connected component, we assign a vertex. So here we have one vertex, here we have one vertex. So here we have in principle just one set in the covering, but it has 2 connected components. So what I'll do is for each connected component, I'll have one vertex. And then when 2 sets intersect, I connect them with an edge. So these 2 sets have an intersection, so I connect them with an edge. These 2 have an intersection, so I connect them, and these 2 and these 2. And then the combinatorial representation that I would get would be this graph that has a loop. And then, so in topology,
How do you pick these nodes in each component? Like in a random way?
No, so you give it. So basically, you're giving a covering with an index set, and then you imagine that that index set is your vertex set.
There are points in the cover that are just abstract.
Yeah.
Abstract.
That's right. Yeah. So this is a graph basically. Yeah. So we have a graph representation of what you see of the underlying space. So now if I took just one vertex for each set of the covering, what I would be doing is taking the nerve of the covering, just sort of for people who know nerves. But this is just different in the sense that instead of taking strictly the nerve of the covering, I first take the connected components and then take the nerve basically on connected components. So then as sort of showed over here, this would be then like a low-dimensional representation of my combinatorial representation of my data set. And then the topological version of mapper, so here is now for topological spaces X and Z, is the following. So we start with a topological space X equipped with a continuous map F to some parameter space Z. So this parameter space will be most of the time the real numbers. And then what we do is we equip this space Z with a covering for some finite index set. And in practice, if we work with Z equals R, it's going to be overlapping intervals. So then, because F is continuous, we can take pre-images of this covering, and we get a covering for X. And then, so then it can of course happen that in these pre-images, some pre-images are going to have, are not going to be connected. So what we do is we take connected components, and then we do this nerve construction on the connected components. And this is then called the mapper of the space. And then, but then of course, so this was sort of the topological version with topological spaces, that's the idea. But with data, I said that the objects are finite metric spaces. So then in this situation, our X is a finite metric space and Z is still the reference map. The reference metric space, and we have a map going from this X into Z. And then we proceed just like before, like we pick a covering for Z, and then we look at the pre-images. And now, question, I guess, for the audience: what happens if we take connected components of pre-images?
Not a question.
Yeah, that's a question.
That's not a question.
It's not a question?
No.
Yeah. Does anyone?
It's not a question.
So, in terms of a representation, yeah, what like what?
Does it look like? Does it have some property?
So, if we have a finite metric space, and then we take these pre-images and connected components, then each vertex is going to be its connected component, and nothing will intersect. So basically what we will get is just going to be the space back, and that's sort of not very interesting. So then the idea is we cannot just take connected components, but we have to take a statistical analog of taking connected components, or the equivalent of taking connected components for finite metric spaces, and that's something that we saw on slide 3, clustering. So the
You left off the word finite. Finite. It's a
covering.
Finite metric space.
Yes.
If you had that, then the question would be.
Yeah, so I think I said in the beginning that the point cloud for me is a finite metric space. That's the sort of definition of a point cloud. And yeah, and the index set is finite, just like before.
It's in the point cloud definition.
Okay, so yeah, so the idea is we proceed like with the topological version, we take pre-images, but then the analog of taking connected components would be to cluster. And then of course here, this depends on the choice of the clustering. And so then once we have clusters, you imagine you put a vertex for every cluster, and then when two clusters intersect, you connect them with an edge. And maybe let's do an example, just sort of, and so yeah, so with clustering, there's sort of, there is a choice to be made, I mean, as it is with the reference maps, but that's sort of also the strength of it. It's like if you select these reference maps in a smart way, then you get information about the underlying data set. So now let's look at this example. So this is a sample from a circle. So just to be clear, it's not sort of like it's not a continuous object. It's like points, so from a noisy circle. So noisy means that so if this is a circle, then you have a sample whose points lie approximately on a circle, but not on the circle. So then we take the filter function to be the distance from the point to P, where P is the leftmost point in the data. So then the space Z would be the real lines, like I said. And then, so then in the mapper procedure, what we have to do is we have to find a covering of this. And what we do is we pick overlapping intervals. So for example, so this would be one interval, this would be one interval, this would be one interval, and here is one interval. So now we have the space, so which is this noisy circle, and we have the reference map F, which is this, and we have Z and the map. And so then what we do is we take pre-images. So for example, if I take this interval, this here will be the pre-image. And then if I take this interval in the pre-image, I have these points, and then here I have these points, pre-image of this, these points, and finally the pre-image of the last one would be these points over here. And now each of these are, again, discrete spaces, discrete finite metric spaces. So if I try to now just take connected components, I would just get all of my points back and there would be no intersections. But what I do is I do clustering. And then when one clusters, so this would be in one cluster and this would be in one cluster. This would be one cluster. This here would be each one cluster. And then what happens is that these two clusters intersect, so we have a connection with an edge between them. And these two clusters intersect, so we have a connection. So we get from having a point sampled with noise from a circle to having a combinatorial representation. And sort of, I guess, so there are some theoretical guarantees that come from the nerve lemma. It's just that they're not always satisfied. But here we are approximating the space. And now I want to go back to the data set that I mentioned at the very beginning. So gene expression data for breast cancer patients. Again, we have a lot of, so for each patient, a very long vector. Each entry says how a gene is expressed. And then, so the goal is, can you, for example, find subtypes of this cancer, like of this cancer? And then, so if this is the data set, then the question is, what kind of a filter function would one use? So in this particular paper, they had so a notion of what it means to be healthy. Sort of, they had, I guess, people that were healthy and they had their gene expression data. So what they did was they took vectors representing the healthy people and they spanned a vector space with those vectors. And then for a patient that, so then if you sort of have any other patient or like represented by a vector, how would you measure deviation from being healthy? So a patient is represented by a vector, and one way to measure deviation is just to take the distance to this subspace spanned by everyone who is healthy. So that's what they did, and that's what they used, this distance to the healthy subspace as the filter function. So then when they produced mapper with Z, the reference space being the reals and some overlapping intervals, this is the image that they got. And one thing that stood out immediately was, so again, this is a situation where we have one cluster, and now what is it, like, why do we have two flares? Is there anything interesting about patients that lie on one of these two flares? It turned out that the blue dots that you see, so this part over here, so this is where all the healthy, I guess, people were. Like, I'm not going to call them patients because they were healthy, but still, it's like in this data, I guess they're all patients. And then over here were women that had breast cancer. But one thing that they noticed was that on this flare, basically everyone so they also had information about the survival rate, and it turned out that on one of these flares, everyone survived, and on the other flare, everyone died. So basically, so why is that important? Because if you can find subtypes of cancer, say, then you can change the treatment based on that and make it sort of more specific. And I guess today everybody is talking about personalized medicine. So this would be sort of a step towards personalized medicine. There is sort of like a name, breast cancer, but that encompasses a lot of different things. And then it's important to know what subtype somebody has, so that you also know what the outlook is and how sort of, you could, for example, change treatment or adapt treatment based on that.
Do you know what year this discovery was made, this particular one?
2010. Yeah, this was sort of one of the, so this was one of the early like nice applications of mapper. Of course, then there were questions. Could you have discovered this by just using clustering, which is sort of like a very traditional, in computer science? And they tried to use just clustering, and basically the subtypes were like completely in separate clusters. So there was no way to do it just by using the standard techniques. Because as you can see, it's one cluster, but it's just that it's different sides of the cluster. And I'm going to show another example of that, actually, which is now about diabetes. So now we're talking about all the important things, all the important diseases of modern world, and why these are important questions. And one of the I guess, pests of the modern sedentary lifestyle is diabetes. So I found this statistics, so like, just 3 days ago, and it's for 2021, 38.4 million people had diabetes in the US, which is 11.6% of the population. So diabetes has been studied for a long time, and it has also been clear for a long time that there are different types of diabetes. And so the most sort of basic division is between juvenile form and the form that one gets later in life. In the 1970s already, there was a study at Stanford with 145 patients, and then they measured for them, for example, age, relative weight, and glucose levels, etc. And in that original paper, this is sort of the picture that they drew, with a central region, and again, two flares sticking out. And then the central, what they found was that these two flares represented the two forms of diabetes. But then, of course, we know now that we can ask further. So if you just focus on the adult form of diabetes, are there any further subtypes that could again be connected to like particular set of symptoms, or like for which you would need like a, like would have so for which you would want to know like so that like that it could lead to some other disease or complications. Then in 2015 they what is it? Icon, like in somewhere on the East Coast. I now forget the name. Econ School of something. So they revisited now this adult form of diabetes, and they wanted to see what they get if they tried to use MAPR on it. What the result was a very nice cover of the Science Translational Medicine journal. So what you can see is, again, a single cluster, but again, so this here and here, but and there are connections between each of these regions, which is why I say that it's a single cluster. But then it turned out that each of these clusters is actually a subtype of diabetes. And then for each subtype, they sort of checked what were sort of the common symptoms, or so for example here, like you have depression, like what is it related to in addition to being diabetes? Yeah, so...
Is this a projection down to the two dimensions?
Yes, yeah. Yeah, so each of these, so the blue is one cluster, the yellow is one cluster, and the pink one, but what's written on it are sort of the complications that one might have with that particular subtype. So these are the two applications that I wanted to show you as far as representing shape goes. And now we're going to move on to the other part of the talk, which is about measuring shape. So in algebraic topology, we often use homotopy or homology groups to measure shape, the shape of topological spaces. The kind of information we get is, for example, if I have a circle, then I can ask, okay, how many pieces is that circle made of? And the answer is going to be, it's just made of one piece. And does the circle have a loop? And it has one loop. Does the circle have a void, like a basketball? No, it doesn't. Then what we get is our numbers associated to spaces that we call Betti numbers. So then if I take a basketball, I can again say, so how many pieces is this basketball made of? Well, it's just made of one piece. Are there any loops? Well, if I sort of look at a loop on the surface, I can always contract it down to a point. And then, the basketball does enclose a void, so it's sort of qualitatively different than, say, a circle. And that's sort of reflected in its Betti2 number being 1. So it has one two-dimensional void. And then, for example, torus is also connected, so it has Betti0, 1. It has two loops, so Betti1 is 2, and it has one void, so Betti2 is 1. And then it's again a question of, well, what we really want to do is we want to analyze data sets, and data sets are finite metric spaces. So now if I use homology directly on a finite metric space, so do I have any loops in that space? I do not. I also don't have any voids in that space. And if I ask about the connected components, what I get for finite metric space is that each point is its own connected component. So basically for Betti0, the information that I get is the number of points. So then it was clear that so homology is a powerful tool, but for finite metric spaces, it just doesn't really reveal information beyond just the number of points. Somehow one had to make changes to adapt this method so it would be appropriate, and this adaptation is called persistent homology. This is a concept that emerged independently, sort of like in various groups. So I guess if you go far enough, you can trace it back to Morse in terms of ideas, but I guess more recently, Frosini, Ferri, and collaborators in Bologna, Italy, Robbins and Colorado. But the most influential paper was by Edelsbrunner, Letcher, and Zamorodian at Duke in North Carolina, Topological Persistence and Simplification. But then Zamorodian went from Duke to do a postdoc at Stanford, and then when he started working with Gunnar Carlson, they actually gave very firm mathematical foundations to this initial theory. So that was sort of then when the math also came into the picture. So now the question is, what does this persistent homology do? How do we adapt our homology method? So we start with a finite metric space, and as said, it doesn't, by itself, it doesn't have very interesting topology. And then one thought is, I guess a little bit maybe in line with what we see over here, that instead of observing just these points, I'm going to thicken each point a little bit. Instead of sort of then observing the points, I'm going to take balls of radius r and the union of these balls. So here what you see, an overlay is already with a simplicial complex that I mentioned earlier, but I will not get into. But that would be sort of the combinatorial structure that you build on top of these unions of balls. So then the question is, okay, so once I thicken these points and take the union, I do get interesting structure. So here I kind of see that, you know, like if I look very closely, that there is a little loop over here. And then if I make a slightly, if I pick a slightly bigger radius, then I see one big loop, very prominent. And then if I make an even bigger radius, I see an even more prominent, like I see, well, I guess less prominent in a way, like, but I see the same loop somehow that I did before, yeah, like, I guess with bigger radii. But then once these radii become big enough, then I don't see my loop anymore. So then the idea is, I could, in principle, approximate my space with these unions, but then the issue I would encounter immediately is that how do I pick my R? Because when I showed you the sequence of pictures, it really depended very much on the R, what I can say about the homology. At the end, like the homology groups were trivial. At the beginning, there were sort of some tiny, like maybe tiny loops, but then somewhere in the middle is what we would, just inspecting the picture, say that's the true shape of the underlying space. But how do you make that formal? Or like how would, yeah, so then the issue is there's no choice of R, and then if we have perturbations in the data, which we often do because of noise, then this invariant is very unstable because you can create new loops very quickly. And then also a related issue is that you cannot distinguish between small holes and big holes. So for example, over here, I have a very small hole, and somehow I want to say that this hole is not significant, but I do want to say that this hole is significant. So then the idea is that instead of taking just a single R, what we do is we observe the entire one-parameter family of these reconstructions, and we have inclusion maps for every pair of spaces whenever R is less than or equal to R prime. And this is called a filtration. And then what we really want to do is we want to track somehow how homology groups change within this filtration. So what we can do is we can apply the homology group in whatever dimension we want to each of these UXRs. Question?
What's the representation that you take of your space pattern? Like instead of taking a circle as a round circle, you take like something that's equivalent to an 8?
So what's the question?
Like if you, the representation that you take of your space when you do this procedure matters. Like if instead of taking a circle, you take something that is equivalent to an 8.
Yes, it matters, and there are, so what I'm describing over here and what you see the picture of is something called a check complex, but then there are other types of complexes that people use, like for example Vietoris-Rips complexes, alpha complexes, so there's sort of like a little bit of an art to doing that as well. And yeah, like you said, it certainly affects. So one nice thing about check complexes is that balls are contractible, and so you actually have conditions satisfied for the nerve lemma, so that the nerve, which is the check complex of this covering, has the same homotopy type as the union of balls. So sort of theoretically, this is then a nice complex to work with because you have these guarantees. But then, of course, there is a trade-off between having theoretical guarantees and also having speed when it comes to computations. So then when it's viewed in that light, then people choose not to work with check complexes as much. But yeah. So we can, so we have this one parameter family, we apply homology groups in whatever dimension we're interested in, and we always take homology groups with field coefficients, just because what we are good at computationally is linear algebra. So if you take homology groups with field coefficients, what you get is vector spaces, and the induced maps are going to be linear maps, represented very conveniently with matrices. And then, so what we end up with is, yeah, as a question? Oh, okay. Is family of vector spaces in linear maps between them. And this R is, so the parameter can be either all reals. In the case that I showed you, it's reals from 0 to infinity. But they are called persistence modules or persistence vector spaces. And then the question is, can we classify these persistence vector spaces? So in general, it's actually a very difficult algebraic question. But we are, at least in the case we are now, which is that we have finite metric spaces and then we build check complexes or like these reconstructions, then we get a very special type of very special persistence vector spaces that are called finitely presented. And it turns out that in that setting, then we actually have a classification, and it is by what we call barcodes. And here, computing persistent homology, as the paper I mentioned earlier, is sort of giving foundations, mathematical foundations to this theory. And so then when I say classified by barcodes, what do I mean? So let's go back to this example. So here we have now reconstructions at these 4 different R values. And so what happens, so what this theorem guarantees is basically so that this so persistence vector space that you get can be decomposed as into a direct sum of what we call interval persistence vector spaces, and each interval corresponds to a topological feature in the filtration. So for example, over here where I have this big loop, so I have one long interval over here. And so what this interval represents is the left endpoint is the time in the filtration when the loop first appears. And then if we look at, so then we have inclusions of space here into this bigger space, and this loop is mapped into the loop at the bigger time. So this loop persists, and it persists across a range of parameters. So then this interval represents basically your loop in the filtration from the starting point to the end point. And what's important is the length of the interval, because the length of the interval is sort of what signifies how important the feature is, with the idea that the longer it is present in the filtration, the more important it is. So the more important it is in the underlying space. So then over here, with this, so what we see is we have a lot of like very, very short intervals, and all of these very short intervals correspond to these tiny loops that formed at one point during our thickening, but also disappeared, or like were filled in immediately. So what you would, I guess, at least like simplistically speaking, say is that what you have over here corresponds to noise, but then you have one very prominent feature, and that is what, and that feature captures the shape of your underlying space. But then, of course, you have statisticians that make that formal and correct. Yeah, I'm sort of building more on intuition now.
Do you have a line for each homology class?
That's right, yeah. So we have an interval. The left endpoint says that's when this homology class first appeared at that time, and then this persists, and it's sort of being basically isomorphically mapped into the next one up until the death time, the right endpoint.
So this is like the Z2 coefficients or something?
That's right, yeah. So usually it's Z2 or Z3, but yeah, most of the time we use Z2 coefficients. So yeah, so over here then in red you can see for these short bars over here, short intervals, the corresponding circles in the filtration. And then here is this loop that I said this is the essential loop. So this is sort of also a way to distinguish then between prominent or essential and non-prominent. With the long bars that sort of, these are the essential, this is the essential feature of your space. Then I will show also just a couple of applications. So one very striking one is to image processing, and so the data set it's a data set of images from the Netherlands collected by Van Hatteren and Van der Schaaf in and around a town called Groningen. And the images look like this, like leaves, like university buildings, a creek. And then, so if you take an image, like a black and white image, you can represent this image as a vector, because so for every so basically a vector living in RP, where P is the number of pixels, because for every pixel you have one grayscale value, so then of course like. These pictures usually have a lot of pixels, so the data set lies in a very, very high dimensional space. And already David Mumford sort of was curious about what can we say about this set of images lying in RP. Can it be modeled as a submanifold of this space? And this is a very difficult question because so the expectation is that the dimension would be extremely high because of everything that you can express. I mean, like everything that is expressed in nature, from leaves to creeks. But then the codimension is also be extremely high because if you sort of take RP and randomly select values, they're not going to represent an image that comes from nature. So then what they looked instead was 3x3 patches. And
3 colors?
No, so within images they looked at, so for example, yeah, so if this is the image P, they would take 3x3 patches, and then they mean-centered the data. They only took like only high contrast images. I mean, there was pre-processing done, but the question...
How many colors were there?
It's just black and white.
Huh?
Black and white.
Well, that's not what you have there.
It is black and white. I mean, grayscale value is given, just one number for each pixel, but the red is just to show the 3x3 square that we are interested in.
So the colors are the grayscale?
Yes.
Maybe between 0 and 256.
That's right, yeah. So this is just to say, this would be an example of a data point.
So black and white means that each pixel is gray, and traditionally gray is measured from 0 to 255.
That's right. Yeah, just
0 being black, 255 being white.
Perfect. Yes, exactly. And the red is just to show this is my data point, 3x3 image patch.
So the 256 colors.
That's right. Yeah. Sorry, when you said color, I was thinking, pink. I think of pink when I think color, not gray.
There's a big difference between what a random 3x3 patch will look like and a structured one.
Yeah.
That would be all solid, or maybe have a stripe in it or something.
Yeah, that's right. So then what was already when Mumford studied these 3x3 patches, he already saw that they lie roughly on a circle. And then the question was, if we use persistent homology, could we try to identify a two-dimensional manifold on which this would lie somehow? And
wait, how did you get to two manifolds? Three-circle method? You're saying the three-circle method?
No. There's no three-circle method. When Mumford observed this data set of, so this is now a data set living in R9. And then what they already found was that the shape is that of a circle. So the reason why people want to look at what sort of model this with manifolds of lower dimension is for compression reasons. Because R9, like you have, it's still living in R9, but then if you want to compress an image, you usually project onto something lower dimensional. So then if you find already, like say, a circle model, that's a projection to a one-dimensional space. So you can sort of use less room. And then the question was, can we find a two-dimensional space on which we could project it would very well approximate this space of patches?
You said two-dimensional space now, and before you said two-dimensional manifold. It's different.
That's true. It's going to turn out to be a manifold.
What?
A model for this space of patches. It's going to be we're going to model it with a two-dimensional manifold.
Where is the data being projected to? What dimensional space?
So the data lies in R9. And
R9.
then
in R9. So we're working in R9 and we might find something two-dimensional.
That's right.
You're saying that Mumford discovered that this data in R9 was
modeled by like
pretty well being populated by a circle, better by a
wait. Okay.
Yeah.
No.
Yeah.
Just asking them.
I want to find out about it.
So the idea was then, they wanted to see if they could sort of find higher dimensional structure that would approximate this space well. So what they did was they basically constructed barcodes on these data points, and so it depended on sort of at what density you observe the image. So Mumford looked at the data set at a different scale than they did over here. And basically, when it was looked at from afar, it had the shape of a circle. And then when they started zooming in, the first thing that appeared was the so-called three-circle model. Where they could actually see so for each circle, you have patches sort of arranged around. So in the so-called primary circle, so this was sort of from Mumford, it was along the diagonal, and then with the secondary circles, the generating patches were all vertical or horizontal patches. And then then the question was, well, okay, so now what about the two-dimensional manifold if we sort of like zoom in really close? And they actually found, so they computed the barcodes for that, and they found, so like the options in the end were either torus or the Klein-Bottle, and then they actually like wrote down like a homeomorphism from Klein-Bottle to the space of these patches to also sort of show how the three-circle model fits into that picture. Klein-Bottle was sort of a low-dimensional approximation for this data set. But why is this? Why would anyone care about this? Here is an image, and now, as I said, the idea is you take sort of these high-density patches, and then you want to find, like, project onto a lower dimensional space. And wedgelet model is based on the circle, and then with Jose Perea and Gunnar Carlson did a compression based on the Klein-Bottle. So I don't know if it's visible, but for example, there are sort of regions where you can see that it works better on the bottom. So, and these are called Kleinlets. So this was...
Which Klein model it is? I mean, there are different ways you can see a Klein model. This is topology without a Klein model.
That's right, yeah. And they actually, yeah, that's right.
It's in 9 space, so.
Yes.
Okay, so there's,
Yeah.
there's only one way.
Yes. Yeah, so this was one application. Another application that I like very much is to the tree of life. So what you're seeing here is from Darwin's book, like his observations of how species evolved. The evolution is modeled by a tree. But of course, Darwin didn't have a lot of the computational equipment that people have today. In molecular biology in particular, there have been a lot of surprising discoveries, like in the late 20th century. For example, in 1977, they found a new domain in life. So they had bacteria as one, for example, as one domain, eukarya, and then archaea was a new domain. What was happening was that when they started observing these life forms that Darwin couldn't observe or know about, it turned out that I'm sorry. I think I skipped one slide. No, sorry. I accidentally deleted one slide earlier when I was making a change, but I will say it. So what happened was that they were looking at viral data, and in particular with viruses, what happens is that you have a virus, and then the evolution is often not linear, because two viruses can recombine and form a new virus. So basically, if you would try, you could try to model this with a tree, but then with viruses, that model no longer works very well, precisely because of these recombinations. A collaboration at Columbia, they gave persistent homology in dimension 1 as a criterion for how non-tree-like evolution is. So that's an application that I like. And then Evita said that there are a lot of pure topologists in the department. So I figured I would also point out that it's not sort of like a one-way process applying topology to analyze data, but it also goes back a little bit. And in particular, there are people who work in symplectic topology who have used a lot of the, who have used persistent homology and a lot of the theorems that proved to be very important for applications. And actually, so here I included those 2 slides from a talk by Polterovich. He has a book with co-authors, Topological Persistence in Geometry and Analysis. So basically, for example, they look at Hamiltonian diffeomorphisms with the Hofer metric, and they have a way of assigning the Fleur persistence module, observe its barcode, and then get a type of a, for example, get that maps are Lipschitz, and then they get lower and upper bounds using persistent homology. So it's sort of, we go from pure math to applied math and back again in a way. And I just want to briefly end with some of the things that I have worked on. I apologize because I'm going a little bit over. So I said that with persistent homology, the structure of the invariant is a collection of intervals. And if you ask computer scientists, they don't like collections of intervals, they like vectors. Everybody wants to work with vectors. And then one big question in our community was, how do we make persistent homology more attractive, so that it wouldn't be, that you wouldn't require too much knowledge in order to be able to use it. Then the idea is, well, we assign somehow vectors to our barcodes in a principled way. And there are two advantages to doing that. So one is, so then you basically represent your shapes. I mean, like you can compare vectors more easily because, for example, in machine learning, you have support vector machine that classifies vectors into groups. So then if you produce persistence diagrams, that can classify, and then if they come from underlying different spaces, you can separate between them in that way. But sometimes the data points themselves, like images, have interesting geometrical structure. And then you might want to assign, say, different barcodes to the same image, and then represent that as a vector that has this shape information. And there are a lot of papers on the topic. For example, persistence landscapes was one of the first papers, persistence images. And so I am in this domain over here. You see tropical beach. So I was, so for me, the goal was to model the space of persistence diagrams. So basically to identify tropical functions that would be symmetric, like if you, because the order of the barcodes doesn't matter. And then also, like for example, if you add a barcode of like an interval of length 0, that shouldn't affect the value. So it was about identifying such type of functions that can be used as coordinates on this space of barcodes, and to prove, for example, that if you wiggle your underlying space a little bit, then the signatures remain reasonably close, so that they are stable, so to speak. And yeah, so then it was sort of necessary to do a little bit of tropical invariant theory, which nobody has done, luckily for me. So yeah, that was, and then we also sort of used that on HIV data. We use some statistical properties like sufficiency, yeah. And then there are other types of problems, like earlier we were looking at this point cloud, and then I said we thicken points. But then, for example, if you know that that your sample comes from a manifold, then you might not want to thicken points the same way in every direction. In fact, like if you thicken them along tangent spaces so that they can model with ellipsoids rather than balls, then what you can do is you can strengthen the signal, the topological signal. And so in some papers, I also worked on that. So for example, here with a noisy circle, much like before, this is standard barcode, and here would be the ellipsoid barcode where these ellipsoids are aligned along tangent spaces. And then of course, with the goal is also that one reduces the size of the sample, so then there are sort of also like all sorts of differential geometry questions that come into the fore. And now I'm working on writing code for this with Bastian Riek and Anna Zegarac, who is a PhD student at ETH. And another sort of current project is, we really like the name of it, it's called Automato. It's a clustering algorithm, an automated clustering algorithm, so that's why the name Automato, where you look at the density function and then do a soup level set filtration, get the persistence diagram, which tells you how to find peaks, and then use statistical bootstrapping in order to separate noise from not noise, so that you get nice regions like in this one over here. And this is joint work with Marius Huber and Patrick Schneider, who are also both at ETH. And yeah, I apologize for the delay, and I want to thank you for your attention.
Any questions?
Well, one of your footnotes said approximating manifolds.
Yeah.
Can
Here.
you say something about that?
Which one?
The homology of manifolds using ellipsoids. Yeah.
Yes. So the idea is that we have a manifold, and then we have a sample from the manifold.
You have a manifold defined by equation somewhere.
It's just a submanifold of Euclidean space, like say Rm.
Any sort.
Yeah. And then.
How is it presented to you?
So it is given to you. I mean, that's why it's.
Assume that, right?
That's right. Yeah. So that's the assumption, sort of, because so the idea is, so what you want to do is, based on this sample from the manifold, you want to. So if I now thicken by balls. So the idea is to say for what range of parameters will sort of this thickening by balls actually capture the homotopy type of the manifold. And you can see already here, if I take a finite sample, that there are going to be restrictions, because if I take the radii that are too small, then what's going to happen is like the homotopy type is just that, like of a discrete space. But then if something that's going to be more obvious, but then if I have something like this, what's going to happen again is, like, if the balls are too big, so if it's too small, then it could be that I don't capture any, like that I don't have enough data basically to reconstruct a manifold. If the balls are too big, then I can have this issue with bottlenecks of this type. For the case of balls, there's a paper by Smale, and it's the Smale that you know, and Weinberger, where they sort of give a range of the R's for which the union of balls, like from the sample, captures the homotopy type. And in the lower bound, this is given sort of by the density. The sample has to be dense enough. And then on the upper bound, it's going to depend on what we call reach. So this would be the distance to the medial axis. So medial axis has points that are equidistant to at least 2 points on the manifold. So for example, here the issue would be that R is bigger than reach. But anyway, so there is an upper and lower bound in the case of balls. And then the question is, if we do ellipsoids that are aligned along these tangent spaces, instead of balls, can we get the same information but have the sample be less dense? Because
all the ellipsoids you drew are supposed to be
along the tangent space.
What?
Along the tangent space. And they're aligned along the tangent space.